Thank you, and thank you to the organizers for inviting me to join you today. It's really a pleasure to be here. I didn't know we were going to have a little North Carolina contingency. So today, I wanted to talk about our project and looking at the use of whole exome sequencing in newborn screening. But in order to do that, I really wanted to give you some basics. I know some of you are probably genetics experts in this room, but others may have very little knowledge of basic genetics. And I think when we talk about some of the new tools that we're able to use in genetics and genomics, it may be helpful to go over some of those things. These are some of the objectives, hopefully, that things that you'll have a better understanding of at the end of this talk. And some of the definitions, genetics just means determined by genes, whereas the genome is really the complete DNA sequence of an individual that contains all of their genetic information. And genomics is the field of genetics that's concerned with structural and functional studies of the genome. So just as a bit of a refresher, if we talk about a cell in the body, any cell except the red blood cells, so any other cells that contain a nucleus contain DNA or genetic information. That DNA is arranged in structures that are called chromosomes. These are tightly wound pieces of DNA in proteins. And then if we were to stretch it out, you'd see the typical double helix making up the DNA. So chromosomes, if you're familiar with cytogenetics, is what this would look like, would represent the chromosomes from one cell in the body looked at under a microscope. And what we do is arrange the chromosomes in order of their size and also based on what's called staining or banding, that is produced when we put a special type of stain on the chromosomes. So they're arranged in order from the largest down to the smallest, and they're in pairs. Chromosomes 1 through 22 are called the autosomes, and then we have the sex chromosomes, the X and the Y chromosome. The chromosomes you can think of being made up of genes on a long string, and each chromosome contains 1,000 or more genes. Now, DNA itself is made up of molecules that are termed nucleotides, and these are the four that you see here. You can just remember A, T, G, and C. And each nucleotide has a corresponding partner that's called its base pair.